Today we're going to be talking about deep learning and chemistry. This is really a field that has opened up in the last, I would say, 4 to 5 years. And yeah, there was a landmark paper in AlphaFold that was developed. And from that point on, momentum in the field of deep learning for chemistry has really picked up. I would say that if I were to partition the decade of the 2010s, the first half of the decade was kind of the time of computer vision with deep learning, where we saw huge improvements on ImageNet, the ImageNet challenge and things like that. The second decade, or the second half of the decade, and I mean, I would say up till today and continuing, has probably been characterized by natural language processing and these models that are able to understand and generate language with indistinguishable accuracy compared to humans. My prediction is that the next decade will be the decade of deep learning for chemistry. And today, hopefully, you'll see some of the reasons for why I believe that is the case. So why is deep learning good for chemistry? So as a first, before I tell you why I think it's good for chemistry, what do you notice about the types of data that deep learning tends to be really good with in your experience?
Deep learning is good for problems with large sets of data and complex patterns that aren't easy for humans to pick up on.
Yeah, exactly. Great. And yeah, that's like basically straight out of the study guide for the midterm, right? So yeah, high dimensional data, large quantities of data, complex patterns. Often there's some sort of structure to the type of data that we're training neural networks on. All of these apply to chemistry. So deep learning is good for regimes with high dimensional data, both small molecules. So we'll talk about what exactly these are in a second, but we'll mostly be dealing with small molecules and large molecules like proteins. And oh yeah, also this lecture will be oriented towards biochemistry as opposed to say organic chemistry or material science, things like that. My experience is in biochemistry, but there are also very exciting things happening in those other fields. Yeah, deep learning is also good for regimes with abundant data. There are 300 million known protein sequences. The space of small molecule graphs is 10 to the 60th, and the space of length 100 protein sequences is 20 to the 100th. So obviously we're dealing with extremely high dimensionality of data for which the true data lies on a very minuscule manifold compared to that gigantic space. There's also this thing called Levinthal's paradox. So I'm calling this chemists discover the curse of dimensionality. So Levinthal's paradox is this idea that these protein molecules, the space of length 100 3D protein conformation. So a 3D conformation is just the 3D coordinates of all the atoms in the protein. The space of these protein conformations is 10 to the 300th. And it would take a time longer than the existence of the known universe to explore all of the given conformations or states of a folded protein. But in our bodies, proteins fold on the pico to nanosecond scale. So yeah, the paradox is that there must be some sort of rule that helps them explore that space of conformations much more quickly than the time of the known universe. So then, yeah, at the same time, something that I have seen is that chemistry actually can really enrich the field of deep learning by introducing new problems that require new architectures and new types of inductive bias. And that's a word that that you may have heard before, but we're going to define in a minute. basically, inductive bias is specific framings of a problem or design of a model that are suited to a particular problem. So there are some kind of unique things about chemistry. We're dealing with problems in 3D Euclidean space, and there are certain symmetries and different types of symmetry in that space. And the question is how we can encode those symmetries into our neural networks. So as a little background on biochemistry, I'm going to give you kind of a rundown on the types of data that we're dealing with. So first of all, we have proteins. Proteins are large molecules in our bodies that perform a variety of functions. They are coded for by DNA and translated from DNA into protein sequences by a molecule called the ribosome, which is itself a protein. Because they are so large compared to other molecules, protein structures are often represented as surfaces, like this image here, or as ribbons, which are easier to visualize than the individual atoms. So there are kind of two representations that you will see for proteins. One is in sequence format. So the way that the proteins work is you can imagine them as different colors of beads on a string. And each of those beads has different chemical characteristics. And we're going to, if we are designing a protein, we're going to select different colors of beads to design a protein sequence that will then fold into a three-dimensional protein structure. And those beads are selected from a vocabulary of 20 possibilities, which we can just represent by characters of the alphabet. Fortunately, there are fewer than 26. So that kind of makes it nice for representing amino acid sequences in terms of letters. And because each of these amino acids is different from each other, these different protein sequences will fold into different folded structures, which then have different functions. Any questions on proteins? All right, so the other type of data we will be dealing with are small molecules. So small molecules are carbon-based molecules, and they tend to be much smaller than proteins. So we represent them, we can represent every atom of a small molecule. However, we do usually drop hydrogens, and we represent carbons as just vertices because there are so many carbons. And so instead of putting a C at every vertex of this molecule, we can just assume that every vertex is a carbon. And so this small molecule is estrogen. So most drugs out there are small molecules, but there are also protein drugs. And yeah, a couple of examples: aspirin, estrogen, dopamine, etc. Okay, so then we're going to be looking at a couple of different biochemistry problems today. One of them is protein structure prediction. Given a protein sequence, we want to predict how it will fold into a three-dimensional structure. What will the 3D coordinates of each of the atoms in this protein be? And this is a ribbon representation of a protein structure. Another problem that we will look at is called ligand-protein docking. So a ligand is just something that binds to a protein, and it's usually going to be a small molecule. And so we're given a protein and given a small molecule, and we want to predict the 3D bound structure of the ligand and the protein. And this is getting very close to what we would call just general drug discovery, which is our third problem. So given a protein, we want to design a ligand, and it could be a small molecule or possibly a smaller protein. We want to design a ligand that will bind to the active site of that protein and prevent some sort of disease that's caused by that protein. Yeah, so one note on that, there are there are useful proteins in the human body, but also the mechanism of disease for many bacteria and viruses is via proteins that attack our body. And so, yeah, a lot of the time when we're looking at dealing with disease, we are looking to inhibit or prevent proteins from doing their job. Hence, we're trying to design ligands that will basically bind to the active side of the protein and plug it up to prevent it from hurting us. Okay, so yeah, I mentioned this word inductive bias. Can you just get a show of hands of if you've heard the term inductive bias before? A couple? Quite a few. So inductive bias is information about a particular problem that the model designer builds into the learning process through, it could be the neural network architecture or the objective function, etc. Oh, and actually, I wasn't supposed to show you that yet. Can you think of anything we've seen so far that is an example of model or architecture with inductive bias? Can you think of an example where we built some sort of real world knowledge about this particular data type into a model? Yeah, Jacob?
I was going to say generative models like GANs, probably.
Generative models like GANs. I'm trying to think how, because GANs are pretty general, right? They can be applied to basically any type of data. So inductive bias is, I'm glad you bring it up because, yeah, inductive bias is kind of particular to the type of data that you're working with. Yeah, and so like there are definitely like interesting things happening in exploring different architectures. Not all of those architectures, though, are architecture choices that are specifically related to a type of data. Tim?
I was thinking possibly RNNs and just the idea that sequence is important in this data that we have.
Yeah, you want to elaborate on that a little bit?
Yeah, I guess just because of the structure of RNNs, we are implicitly telling the model that our data belongs in a specific sequence that we give it. And so that we can't get the same results if we change up that sequence. I don't know if I'm explaining.
Totally, yeah. It's kind of taking advantage of the ordering of sequences in RNNs. Yeah. Another kind of the classic example of inductive bias is in CNNs. And maybe you were being kind and forgiving my and not bringing up CNNs because you didn't want to cheat. I appreciate that. But yeah, so CNNs are a great example of inductive bias where the bias that we are taking advantage of in the data set is this idea of spatial locality, that positions close to each other in the tensor have more to do with each other than positions that are far apart from each other in the tensor. So we're going to define these convolutions, these convolutional filters that are only allowed to look at a small spatial window in the tensor and basically have an enforced sparsity pattern where they're not allowed to look at anything else in the image. And so, yeah, in that way, CNNs, we're kind of telling them that you should be looking for features in these spatial groupings of pixels. Does that make sense how CNNs use inductive bias? Okay. So what are some types of inductive bias that exist in protein folding? So it turns out that for protein folding, there are a lot of constraints. So naively, maybe something you might do is just design it. Well, actually, yeah. If you were to design a neural network that takes as input a sequence of characters and predicts 3D coordinates for each of those characters, what architecture might you use? You were tasked with building a protein structure prediction model, a protein folding model. I'm going to take as input a sequence of characters and output a sequence of coordinates. Yeah, Brandon, what do you think?
An RNN?
An RNN, yeah. So the RNN would take as input the sequence, and then So you're going to have 3D coordinates for the output, right? So you just have the output vector of the RNN be a vector of length 3 for each position. Yeah, and so then so remember we talked about 3 different types, actually 4 different types of RNNs. There was a one-to-one, a many-to-one, a one-to-many, and a many-to-many RNN, right? Do you guys remember that? And then in particular, there were 2 types of many-to-many. There were 2 types of many-to-many RNNs. Does anyone remember those 2 types? Yeah, Spencer?
I think it was just scale up and scale down, like going from 2 inputs to 10 or 10 inputs to 2.
So not exactly, but good shot. So that's kind of like many to one versus one to many. What I was kind of going for is there are many to many networks where the input and output have different lengths from each other. Like in the case of machine translation. However, if we want to do like part of speech tagging, we're going to have one output for each one of our inputs, right? And so those are the 2 many to many tasks. One where you have different length input and output, and one where you have one output corresponding to each input. So with that in mind, Brandon, which of those do you think you would want to use for protein folding? Yeah, the options are predicting one output for each input versus predicting a variable length of a different number of outputs for a different number of inputs. The second one, why would you do that? Do the second one? Gotcha. Interesting. So yeah, I'm not sure exactly how it would work if you had a different number of inputs and outputs. I would probably do it as the many-to-many problem where you have one output for each input and you're trying to predict 3D coordinates for each character in the input sequence. Does that kind of make sense? How we're going to set up that many-to-many problem? More or less? I feel like I'm losing you guys. Are you guys not interested in chemistry? A bunch of computer scientists. All right. Yeah, what do you think? Hey, yeah, ask a question. I'd like some help here to help you guys.
Remind me the question.
Well, so where are you lost? Or
why particular?
So no, sorry, we're not talking about CNNs for this problem. We're asking the question of what architecture we might use for a protein structure prediction problem. So yeah, what do you think?
Yeah, so we talked about using RNNs for it, and then the type of RNN that we'd probably want to use is the many-to-many with non-variable length, like this fixed input and output, because well, just because the way like proteins are structured, they're not going to like, you're going to see one result for one input. You're going to, because there's only like one type of like folding, 3D folding that you will typically see for one input, so you don't want variable length. So that's probably the type of architecture you implement.
Yeah, exactly. That's probably how I would choose to do it as well. Yeah, what do you think?
I was thinking a transformer, like kind of like for the language translation, because you have the sequence of inputs that get converted into like one condensed, right? And if you could somehow just take that condensed version and turn it into the folding without kind of taking it out token by token, like all the information is there, right? Something like that.
Interesting. So are you describing a transformer encoder, a transformer decoder, or a transformer encoder decoder?
Something, just the encoder, and then you'd have something different for the decoder because you don't want it coming out in tokens. You want it coming out as like one thing.
Gotcha. Interesting. So I like the way that you're thinking. So I like the idea of using a transformer to handle the variable length input. We do want so we're trying to predict a variable length output, right? We're trying to predict one coordinate for each input token, right? So you're right though that we're predicting coordinates. We are not predicting tokens. right? And so perhaps maybe a variation on that we could, or does anyone have it, maybe an idea, or even you? Yeah, predict them all at once. So yeah, a transformer decoder actually does exactly that, right? You take and each token is input, and then you predict one output for each of the, actually either an encoder or a decoder could work, where you predict one output for each of your inputs, right? So you have a transformer encoder that takes as input a protein sequence and provides as output predictions for each of the coordinates in that protein sequence. Does that make sense? Yeah, Jared.
So I might be misremembering transformers a little bit, the decoder a little bit. In this problem, I assume you would want it to be able to input variable length sequences. It doesn't have to be the same length every time, but that would also mean your output has to be a different size every time. And wouldn't decoders have trouble with that? They want to have the fixed output size every time.
So both transformer encoders and decoders can have variable length input and output. And the length of the output corresponds to the length of the input in transformers. And that was the exercise that we did a couple of weeks, or yeah, when we did the transformers, where we traced the dimensions and we saw that the outputs, the length dimension of the outputs corresponded to the length dimension of the inputs. Yeah, I really like these thoughts. So I think we've come to a pretty good starting point of how we would design a protein structure prediction model without any knowledge of chemistry. However, the key idea here is that we're going to try to take some knowledge about how proteins are and the type of data and try to make the learning problem easier for our network. So here are some facts that I'm going to tell you about proteins. So number one, bond lengths are constrained. So there is a fixed distance between each amino acid. So already we've now, instead of predicting just random coordinates, I've told you that basically the next coordinate falls within a sphere of fixed length around the first coordinate that we predicted. Does that make sense? So we have constrained bond lengths, constrained bond angles as well. And I should have had a diagram of this to explain the difference between bond angles and torsion angles. But basically, if you imagine a tetrahedral molecule. So you have a atom at the center, and you have 4 spokes coming out of that center atom. That's how most atoms form bonds. So you have the central atom and then 4 spokes coming out, and the angles between any of those are pretty rigid. However, if we introduce another atom into this, and so each of these atoms are bonded to each other, and then they're also each bonded to 3 others, there is a degree of rotation around that axis by which they are attached to each other. And that rotation around a bond is called a torsion, or a torsion angle. And so the angles between each of the bonds is not changing, but the rotation around those bonds is changing. And so between these 2 constraints on bond lengths and bond angles, the degree of freedom of protein folding are actually very limited. Okay, so that's some amount of information for you. I'm also going to tell you that the protein folding function has the property of equivariance. So equivariance is a mathematical property where it says that f of g of x is equal to g of f of x. And protein folding has equivariance to 3D rotations and translations. And so what this would look like is folding of a 3D rototranslation of our input coordinates is equal to a 3D rototranslation of the folding of our input coordinates. Basically, the way you can think of that is if we take as input to our model 3D coordinates and we rotate them, then we want the neural network to rotate its predictions by the same amount that the inputs were rotated. So, and that's honestly probably the hardest part of this problem. How do you, how might you, if you wanted to train a neural network to be able to predict protein folding, with any rotation of its input coordinates, how might you do that? So I'm going to, and before you tell me that, I'm going to propose a modification to our original architecture. So now our original architecture, instead of taking just a sequence as input, it's going to also take as input initial coordinates, and then it's going to update those, then it's going to output updates for all of those coordinates. So now our network takes as input coordinates and then produces coordinates as output. So if you had a model like that, how might you try to train it to respect all possible rotations of those input coordinates? Good.
I'm not sure if I'm understanding this perfectly, but it sounds like a data augmentation sort of problem. So you would augment, you have, I don't know how many, 140,000 data protein structures or something like that. And you could augment all of them with all the possible rotations. And then you'd have to train the model to predict like any of these are correct answers. You could predict any of them.
I am so glad that you said that. That is like that is probably what we would have said like 5 years ago in deep learning. It's like but that's totally a valid solution. Yeah, provide the network with every possible rotation and translation of the input coordinates, and basically kind of by brute force give it enough data to be able to learn that equivariance property. Spencer.
Couldn't we just throw in a function that converts all your Cartesian coordinates to spherical coordinates? And then no matter what rotation happens, like you've made your own like internal coordinate system that doesn't care.
Totally, man, you guys are so smart. that is what people end up doing. So what Spencer is proposing is basically to choose an internal coordinate frame, and instead of predicting absolute coordinates, predict relative rotations via spherical coordinates for each successive atom in this chain. The benefits of that are that instead of having to have a massive data set to try to learn this equivariance property, the equivariance property is built into your neural network. Let me repeat that. So instead of learning the equivariance property is built into your neural network. Your neural network can only learn functions that are equivariant to rotations of the input. Any ideas why that might be helpful? Why we would want it to be built in instead of learning it?
Like basically that constrains like your solution space. And when you're dealing with something that's several orders of magnitude, reducing that order of magnitude by even 1 or 2 just with a certain constraint vastly increases, like it improves your speed and everything of finding actual solutions instead of finding a bunch of bad solutions because it wasn't constrained enough to begin with.
Exactly. Yeah. And so yeah, you said it perfectly. Basically, instead of our neural network wasting time learning a property, wasting parameter values and wasting time, like learning properties that we can build into the network, we just build them in, and then it can spend its learning capacity on learning the actual pattern we are looking for, which is the folding. Okay. So this is AlphaFold. it has two subparts. It first has an embedding module, which is a transformer with axial attention. The way that axial attention basically works, so it takes as input a protein sequence, and then it also takes as input what we are going to call a pair representation, so the pair representation is L by L, and then the protein sequence representation is L by N. So I told you it takes as input a single protein sequence. It actually takes as input a multiple sequence alignment between the original protein sequence and evolutionarily related sequences. So maybe the ribosome sequence from a human and then the ribosome sequence from a fish, for example. And so this is kind of interesting because when we saw text only has a sequence length in one dimension, but here we have sequence length basically in two dimensions, the length of the amino acid sequence and the number of sequences in the alignment. And so there's an architecture called axial attention that basically performs attention across sequence length, across one dimension, and then across the other dimension, alternating back and forth. Does that kind of make sense how that would work, axial attention? Okay. And then I won't go a ton into the pair representation. Basically, you can think of this as representing interatomic distances. So yeah, that's our embedding module, which spits out a representation that has been learned for the protein sequence. Then the second part is an SE3, which SE3 is the mathematical group of rotations and translations. And they use an SE3 equivariant graph neural network to represent those relative rotations and translations that Spencer was talking about. They don't use polar coordinates, but we are going to discuss actually how that might be done. So you suggested predicting rotations as polar coordinates. Can you guys think in like the context of deep learning, why might polar coordinates be a difficult thing for a neural network to learn to predict? Because they're continuous. I'm going to say not exactly, because we predict lots of continuous values. It's called regression. You can predict someone's height or weight, and neural networks tend to do pretty well at that. Is a good thought, though. Yeah, go ahead.
Is it because you might use sinusoids to calculate like what you actually want when you're using polar? Like regular ones, you can just add them. Like
yeah. So you're getting at a really important idea, and that is that often we're most interested in the sine and cosine of the angle rather than the angle itself. So yeah, in the case of the real problem is that polar coordinates are periodic. So spherical coordinates, you have basically three angles that you're predicting, but in terms of training signal, 2 pi is the same as 0, right? And so if we're trying to get a gradient to change our prediction of that angle, it's difficult for the network to handle that periodicity. So one option is you might instead predict the sine and cosine of an angle, and those are more continuously valued. Yeah, any other thoughts of how you might represent a rotation in a prediction by a neural network?
Could you restrict the domain from 0 to 2 pi using a transformation?
Restrict the domain? Yes. Yeah. So you could totally use like modulo. Apply the modulo operator so that you can only predict things between 0 and 2 pi. But you still have the periodicity where basically your loss function is discontinuous at that 2 pi. I see. So again, you're suggesting predicting a yeah. So yeah, you're recommending, yeah, applying some sort of nonlinearity that restricts our domain. But you still have the issue of the discontinuity of the loss function, because say your network gets to be predicting like 2 pi minus 0.01. Yeah, and so, but it's a good thought. Yeah, you definitely would want to restrict the domain. Any other ideas of how you might predict a rotation from a neural network? Any ideas of how do we normally mathematically represent rotations? We can use polar coordinates. Have you guys taken linear algebra? Any ideas? Yeah.
A rotation matrice?
Totally. Yeah, you could predict a rotation matrix. Yeah, rotation matrix. So a 3x3 matrix can represent a rotation in 3D space. I think that's probably how I would have done it. It turns out though that a 3x3 matrix actually has more degrees of freedom than actually exist in a rotation. So a rotation only has 3 degrees of freedom. Granted, you can also 3x3 matrix can also represent a translation. So there are really 6 degrees of freedom that we're looking for. 3 translational degrees of freedom, X, Y, and Z for translation, as well as X, Y, and Z, or as well as 3, you can think of them in polar coordinates of rotation in 3D space. Turns out that what they use are these things called quaternions. Anyone ever heard of a quaternion? Yeah, so quaternions are basically a way to represent a 3D rotation as a vector of length 3. Technically a vector of length 4, but there are only 3 degrees of freedom in that vector. And it turns out that these have really nice optimization properties. And so, yeah, the way that they predict their relative rotations are in terms of quaternions. How do they do? Well, this is a slide from the first day of class. This is in the semi-annual competition on protein structure prediction. And this tall bar at the left is AlphaFold, and all of these are everyone else. This graph, the graph on the bottom right, shows basically maximum experimental accuracy of about 93%. That's like the best you can do by experimental methods at capturing a protein structure. And AlphaFold 2 gets up to about 90%. So really a huge leap in biology. And nowadays AlphaFold is being used widely by basically everyone in biology and biochemistry. And yeah, AlphaFold is able to predict protein structures of up to 2,000 amino acids. So this is an overlay of a true structure given in green and a predicted structure in blue. And you can see basically that there's almost perfect correspondence, at least except in a couple of boundary areas, between this extremely complicated experimental protein and the prediction of AlphaFold. Any comments or questions on that? Yeah, so this is kind of woke the chemistry world up to the idea of how powerful deep learning can be for chemistry problems. We're not going to go a ton into this one, but this is a recent paper that tackles the problem of protein ligand docking, which, if you remember, is the problem of taking a protein and a small molecule and finding the pose and orientation by which the ligand will bind to the protein. And the way that they do it is they define a diffusion process using it, and they use a diffusion model over translations, rotations, and torsion angles. So the translations and rotations are the small molecule relative to the protein, and the torsion angles are the degrees of freedom in the small molecule. So if we look here, anywhere that isn't anchored by any bond that isn't anchored by 3 atoms is fair game for a torsion. So that you can have a torsion here where you're twisting these two hexagons. You can also have a torsion here where you're twisting these two hexagons. And so, yeah, we're basically predicting a rototranslation and torsion angles of a ligand relative to a protein that will bring the ligand to the bound location on the protein. And yeah, this just describes those degrees of freedom. And long story short, they do amazingly well and basically double the accuracy of previous docking methods for, of even deep learning based docking methods. So yeah, if you're interested in deep learning for chemistry and the problems that are out there, feel free to talk to me. I'm always happy to hype it up and try to convince you to change your career path. So yeah, have a great day and we'll see you on Wednesday.